the recording. All right. Hello, everyone. I welcome all of you on ISNM MISSI webinar series. It's a monthly session, so the talk is going to be around 45 minutes, followed by 15 to 20 minutes Q&A session. During the talk, I would request all of you to mute your phones and devices, and then we'll take the questions at the end of the talk. Feel free to put your questions in the chat and we'll address them after the talk. Now it's my absolute pleasure and honor to introduce Dr. Avinash Eranki, who is currently an assistant professor in the Department of Biomedical Engineering at Indian Institute of Technology, Hyderabad, India. And he directs a medical ultrasound research lab that focuses on developing novel ultrasound imaging and therapy options for solid tumors, such as breast and liver tumors. The lab also develops novel algorithms and sensors for image-guided ultrasound therapy and novel genomic work to supplement the effort to better understand and treat these tumors. Dr. Eranki is also the Chief Executive Officer at the Eranki Labs Private Limited. It develops and manufactures high-intensity focused ultrasound systems for treating solid tumors. He received his undergrad degree in electrical engineering from Bangalore, India, and Masters of Science in Electrical Engineering from George Mason University, Fairfax, Virginia, USA. He then went on and did Ph.D. in medical imaging / therapy from Utrecht University from Netherlands. Before this current position, he was a research and staff development engineer at Children's National Medical Center and Washington, D.C., and a research collaborator at the Department of Interventional Radiology at National Institute of Health Sciences, Bethesda, Maryland, from 2014 through 2020. Without further ado, Dr. Eranki.
Thank you very much, Dr. Saima. Thank you, everybody, for this opportunity. Very briefly, we work on ultrasound imaging and therapy for solid tumors. Just give you a very quick background on what our lab does. So we build devices that are towards image-guided ultrasound therapy for different solid tumors. We've started out with breast tumors, already getting into liver and potentially others as well, such as sarcomas. We also have a few trials that are ongoing for imaging, and I'll talk a little bit of that and show you some data on that aspects, and coming up with new ways using ultrasound imaging and quantitative approaches to see if we can pick up features in tumors that are otherwise not picked up right now. We also perform acoustic modeling. So we try to understand how waves propagate in different tissue types, both the longitudinal waves and shear waves, what kind of shear stresses you can expect if there are changes in collagen content in a particular tissue type, which is generally not very uncommon in certain heterogeneous tumor types. We also make our own novel sensors that are then combined and implemented on our HIFU system. These sensors are used to detect temperatures, pressures, along with a couple of other markers. We also have done a little bit of work, very preliminary right now, a short clinical trial on genomics and liquid biopsy in women suffering from triple-negative breast cancer. And we also give back to the community. We build certain devices and have built software that go along with it that we give away to certain members of our medical community in India that otherwise cannot afford things like 3D ultrasound imaging and so on and so forth. These things are often not patented or published, and we just do that out of what we feel is the bare minimum we can for the community. So I just start with where the whole need to treat solid tumors in different ways. And there's a couple of challenges that we continue to face in terms of certain tumors that tend to be very cold or non-immunogenic, very little infiltration of tumor and immune cell types, and the insufficient or lack of insufficient or lack of tumor-associated antigen repertoire, And so, your stromal layer formation on tumors, certain tumors like pancreatic, which tend to show a lot more than others. And a lot of the T cell pathways tend to be very regulatory. There's other causes as well. This is definitely not an absolute exclusive, but um, some of these or a combination of these is just pretty much good enough that even multimodal therapies don't clearly work. And so there's a clear unmet need to make some of these tumors hot and sensitive to current drug therapies. So the way we sort of work right now is a device drug combo to improve tumor-free survival, systemic cytokine and cellular response, and change the genetic variants that you otherwise do not see, and release tumor-associated antigens in the milieu of the tumor, as well as in the draining lymph nodes and the contralateral lymph nodes, and hopefully elsewhere that could get attention from your antigen picking up cells like dendritic cells and then eventually T cells. So the whole field of MR and ultrasound guided HIFU is right now you have two clear classes of HIFU or high intensity focused ultrasound, or HIFU. Is under MRI guidance, MRI imaging guidance, and this is an image from a Philips magnet and an originally Philips designed system, but is now owned by Profound Medical, based off in Toronto, Canada. These two systems are examples of ultrasound image guided HIFU systems, a very popular prostate HIFU system under ultrasound imaging guidance. And this is a relatively newer system by Theracleon, a company based off in France, is currently running a breast fibroadenoma trial, or I think they must have just completed one at UVA. Using ultrasound imaging guidance. And so there are pros and cons to either of them, but eventually these are currently in the market and doing reasonably well. So HIFU or high intensity focused ultrasound has a few flavors, and one of the key flavors is to thermally ablate. And what we found is that with thermal ablation that we saw a certain type of response. We then said to increase the antigen availability in the microenvironment of the tumor, as well as in the draining lymph nodes and contralateral lymph nodes. It may make sense to mechanically fractionate some of that tumor tissue. So one of the ways that we started developing that is using something called as histotripsy, and specifically speaking, boiling histotripsy. And so that's based off on shocks that you create. What you're looking at here is data that we collected on the clinical ultrasound system, which is the MR guided HIFU system, or the Sonaleave system by the Philips. And what we measure is pressure in megapascals in the y-axis, and the x-axis is time in microseconds. And as we sort of vary the power, the acoustic power from 500 watts to 550, 600, and 650, we see an increase in the acoustic pressure. Now, what this simply translates into is every ultrasound wave is in some sense a compression and a rarefaction. It's a mechanical wave. But anytime you see a shock that's developed, so that means that when you go from your peak negative pressure or peak rarefaction pressure all the way to the peak positive pressure or your peak compression very quickly, it's almost like a very strong and fast punch. And then you slowly retract back when you go from the peak positive pressure all the way to the peak negative pressure. This tends to sort of do a couple of different things. And one of the things it does is very quickly starts boiling the tissue in a certain region. What you're looking at is multiple of these cycles for a period of anywhere between 2 to 10 milliseconds. And you repeat that every 1 to 2 seconds. And within the 2 to 10 milliseconds, there is a time called the time to boil. And it takes a certain time to boil, and the tissue inside starts boiling. Now, this is very, very spatially localized to the tune of few hundreds of microns or maybe a millimeter or two. So it does not spread to the surrounding tissue. So what that eventually does is it causes rapid and localized tissue fractionation with millimeter precision. And I'll show you a little bit of data on that, as well as leaving the surrounding microvasculature and tissues viable. So what you're looking at is liver tissue that was treated with boiling histotripsy. This is about 3 millimeters, and this is about 5 millimeters, which is what we had planned. These are liver lobules, hexagonal shaped, that are completely intact in the surrounding region of the targeted boiling histotripsy lesion, as well as viable. So one way we measured viability was to look at the mesenchymal cell origin or stain. Sorry about that. Yeah, sorry. So what we saw is once you close up a little bit, that it does stain very well and it stays intact. And as you can see, the boundary of the tissue continues to stay completely intact. While the tumor region that was treated with boiling histotripsy has fractionated tumor debris with antigens in that region released in the milieu. So what we showed is that you can perform this in multiple spatial locations in the tumor. Especially becomes very cool and interesting when you have a heterogeneous tumor like triple negative breast cancer. It's completely non-invasive, uses non-ionizing radiation, and it's performed as an outpatient procedure. A lot of patients that we treat across the world in different centers using HIFU are pretty much a day procedure at most. We've also seen that it leads to rapid sterile inflammation and the release of DAMs. DAMs stand for danger-associated molecular patterns. And the sterile inflammation, in some sense, is key to produce a strong immunogenic response against the tumor and to create an anti-tumor T-cell response. And some of our data also shows that it does not lead to tumor metastases, and this is something that others have also demonstrated, and now very recently even in clinical trials, using a slightly different flavor of HIFU. And so that makes it very exciting, because one of the key questions were, if you were to fractionate a cell, are you going to create more mets? And we think no, from preclinical and clinical data we currently have. So what we did with the HEMR HIFU system is we varied the parameters to understand the degree of temperature that went to the surrounding tissue because of those boiling bubbles that were created. So we used MR thermometry, and we looked at the center of the region of interest that we treated, or the center of the focal zone of the ultrasound therapy, along the coronal and the sagittal, as well as we placed a second slice that was a couple of meters away from it. And so if you looked at the temperatures, temperature maps that we received with the temperature in degrees in Celsius in the y-axis and time in seconds in the x-axis, at the focus, you did have an increase in temperature that exceeded your thermally ablative temperatures. But a couple millimeters away, the temperatures were well below 50 degrees Celsius with this set of parameters. If you change the parameters, increasing the time of your sonications, although the temperatures at the focus were ablative in nature, at about a couple millimeters away, you stayed well within the mild hypothermia temperature. Now, this made it very exciting because it's in this region we could potentially release certain drugs that were temperature sensitive. A great example is Thermodox, which is a temperature sensitive liposomal formulation to encapsulate doxorubicin. And that generally gets activated anywhere between 41 to 43 degrees Celsius, the greatest being at around 43 degrees Celsius. So all we're saying is that we can mechanically fractionate tissue at the center and release drugs selectively in the tissue that surrounds the treatment region, which is intact and viable. So it's kind of two bullets to the same bird. So we then also looked a little bit under MR. So this is a piece of liver tissue And this is T1 and this is T2 weighted. This is along the sagittal and this is an axial slice. So this first column is an untreated piece of liver tissue, and this is post-treatment with boiling histotripsy, where we actually show a lesion that is in the form of a tadpole shape, which is what you expect. You always expect the lesions to be a little longer along the ultrasound beam axis and a little shorter along the across the ultrasound beam axis. You also see a hyperintense signal in the axial direction in these tissues, suggesting tissue being completely liquefied. These are subtracted images. So essentially, we subtracted B minus A and E minus D, and that kind of improves our contrast to noise ratio, so the ability to visualize the lesions. So under MR imaging, we're also able to visualize the lesions after treating them. We then said, if we vary the parameters a little bit, does that change the degree of fractionation? Or does it change the amount of temperature change, or does it do both? So the top panel here are thermometry maps under using MRI, PRFS. The second one is gross pathology, and these are H&E stains. With a certain set of parameters, we saw a greater increase in temperature, closer to 60 and maybe a little beyond. And we also observed some fractionation of liver tissue. All of this was liver tissue. You also see a little bit of whitening of tissue around this region, around this border of this green dotted line, suggesting thermal denaturation of tissue, possibly because of conductive effects in that region. So that means that we were able to fractionate as well as heat up that region and also the surrounding region. A second set of parameters, the temperature barely went very close to 50 degrees Celsius, maybe 1 or 2 voxels at most, but pretty much stayed around the mild hypothermia region. But the tissue was completely liquefied with the surrounding tissue intact and did not show any signs of denaturation. And the third set of parameters, the temperature did go up a little more, but we punched a hole completely through. All was left over was a hole through and a little bit of liquefied debris. The borders were still sharp, and we didn't see any denaturation of tissue. So that was exciting because the parameter space is so broad, it's pretty much an ocean of parameters. But our ability to sort of maybe pick a few from that menu, depending on the tissue type, helps us decide whether you want complete fractionation or a combination of a little heat. So that's sort of what we set out to do with this. Then we said, does histotripsy sensitize a systemic immune response and improve survival? So we took a murine model of neuroblastoma, and the tumors were refractory. The diameter of these tumors were greater than 10 millimeters, so oftentimes very close to 12 or 13 millimeters using the calipers. And we treated them with HIFU using a preclinical setup that's seen here under ultrasound imaging guidance. We used a positioning system, as seen here, is an XYZ positioning system, computer-controlled positioning system that ensures that the beam is targeted in the right location every time, and the foci are positioned as accurately as possible. This is an H&E of the untreated neuroblastoma tumor. And post-boiling histotripsy, we see, again, results that were otherwise seen in ex vivo tissue, where we found debris of fractionated tumor tissue, and the surrounding tissue shown by the yellow arrowhead was completely intact tumor tissue. And very interestingly, treating them with HIFU and immunotherapy completely treats them, and the tumor disappears and regresses. So this was the tumor before. It's the same mouse with the tumor after. And that effect stays for at least 300 days, which otherwise is not seen with boiling histotrypsy alone or boiling histotrypsy with anti-PDL1 or anti-CTLA4. So you can either use atezo or EP with boiling histotrypsy did not show us a great response. But when you combined boiling histotrypsy with EP and atezo, it seems to do much better. And this was significant compared to every other group. What then sort of indirectly suggested is a lot of these tumors were fairly refractory and could not otherwise be treated or cured. Obviously, if you left the mice untreated, they all died very, very quickly. So we then set out to see if we could understand that a little bit. So we treated these mice again with HIFU only. So we had a few mice that were untreated, seen in the first column, treated mice with HIFU in the subsequent 3 columns. We euthanized them at 24 hours, 48 hours, and 72 hours. And we looked at the tumor tissue. What was immediately obvious is that you had a few resident macrophages. CD68 positive stains is a pan macrophage marker, so stains for macrophages in general and a couple of other cell types. So we think there were some resident macrophages which seem to increase a little bit in 24 and 48 hours before it slowly starts coming down. We did not observe any CD4 positive or CD8 alpha positive T cells, suggesting that these tumors are quite refractory in nature and cold, so there was no infiltrates of T cells of any sort. But 24 hours after therapy, there was a significant increase in the helper T cells, as well as a bit of a jump in the CD8 alpha positive cells. And then very quickly, that effect dies down by the time you reach day 3, possibly suggesting why, if you treated these mice only with HIFU, they did not survive. So there was something going on in this initial time frame that holds the key towards improved tumor regression and survival. We also looked at the spleen, the draining lymph node, and the contralateral lymph nodes. This figure here shows the different cell types that were measured. So DXY is a marker for natural killer cells. We saw a significant increase in the natural or NK cells 24 hours after treating. The first row are all untreated mice. And then we have a 24-hour, 48-hour, and 72-hour. So in the acute phase, there was a significant jump and again, a significant drop back in the innate immunity, both in the spleen and in the draining lymph node. We also saw a bit of a jump, an increase, but nothing significant in the CD11C positive cells, which is a marker for dendritic cells, as well as an increase of the same cell type in the draining lymph nodes. We also saw a significant increase in the CD8 alpha positive dendritic cells, which are known to be more or have a greater propensity for an anti-tumor response in terms of performing greater antigen presentation, both in the spleen and in the draining lymph nodes. The regulatory T cells, the CD4 positive FOXP3 in the spleen and the draining lymph nodes, significantly went down over the period of 3 days, which was also very encouraging, suggesting that there might be a chance that the ratio of your CD8 alpha positive cells to FOXP3 in the spleen and draining lymph nodes increased significantly, hopefully tilting the entire response towards a more cytotoxic T cell pathway and not a regulatory T cell pathway response. We also sampled systemic blood via cardiac puncture, and we saw an increase in certain cytokines and decrease in some others. The y-axis is the concentration in picograms per milliliter at 3 different time points, same as before. We saw a significant increase in IL-2, which is suggestive of pro-inflammatory response, and IL-2 is also a cytokine that drives NK and cytotoxic lymphocytes. We also saw a significant reduction in VEGF-A, which was very encouraging, in addition to IL-10, which is a more of an anti-inflammatory response, and IL-10 is known to drive TGF-beta and its associated response, possibly creating a more tumor therapy response, or in other words, pro-tumoral response. We also saw an increase in GM-CSF and certain fluctuations in IL-6. What was most attractive to us at that time was interferon gamma. We did know that immediately after therapy, in a few hours, there was a significant inflammation, but that was not seen or observed in the blood concentration of interferon gamma. It was very surprising because that's sort of what we expected. But it did shoot up on day 3, which we said, that sounds now about right. So then we set out to see if we can understand what happened there. So before we tried to understand the effect of interferon gamma, we then said, what else does interferon gamma do? Here's a stain, a DAPI with PDL1 stain in untreated tumors. And what you actually see is a very cold tumor with no PDL1 expression on the tumor cell surface of these neuroblastoma, murine neuroblastoma tumors. But after treating them with HIFU and at 72 hours, you saw a significant increase in PDL1 expression on these tumor cells. And that was very exciting. And we knew this was driven by interferon gamma based on some of our in vitro experiments, suggesting that PDL1 upregulation was due to interferon gamma. So in an effort to understand what was going on with interferon gamma, as well as what do you generally expect in the genetic landscape, we did a microarray for mice that were untreated, treated at 24 hours, 48, and 72 hours. And we had 3 housekeeping genes, GAPDH, ACTB, and GUSB, all 3 confirmed to not change to any form of HIFU therapy. We saw a few DAMs that were that had a significant fold change, S100A8, A9 heat shock proteins, 27 kilodalton as well as the 70 kilodalton, as well as HMGB1. And then we also saw LCN2 as well as CD72. One of the things that we observed was some of these genes had the ability to suppress interferon gamma production by cells. And once CD72 went down in terms of its expression, interferon gamma went up. So this at least was some clue to why we did not see an immediate jump in interferon gamma while we saw that for IL-2, for example, in the blood. So then we were very interested in an abscopal effect. Obviously, we observed systemic immune response in a certain pattern, and we were very curious to see if that really did anything good systemically. So again, took the same mice, same tumor type, neuroblastoma, grew 10 millimeter tumors on either limbs or hind limbs, and we only treated one. So we shaved the hair, the other one remains unshaved, and we treated one. And the tumor completely regressed on both sides. So not just on the treated side, but as well as on the untreated side. And we had 18 such mice that we treated that survived for 300 days, or a fraction of them survived for about 300 days without any tumors. So we initially had a few losses, but then they consistently held after that. But if you used only immunotherapy, which is your anti-CTLA4 and anti-PDL1, none of them, except one mouse, none of them survived. So that to us said, well, the immunotherapy does seem to do something. but the combination seems to have a significant effect. So this could suggest that HIFU acts as a strong adjuvant to your systemic immunotherapy. Mice that actually survived HIFU or histotripsy with immunotherapy, we isolated their splenocytes, and we wanted to look at what was going on. So after these mice survived for about 300 days, we re-inoculated them with 2x of the initial tumor inoculation. So initial tumor inoculation was 1 million cells. After they survived for 300 plus days, we re-inoculated them with 2 million cells. And none of those mice grew tumors. 100% of them continued to survive. Both mice that had a single-sided tumor as well as two-sided tumors for greater than 65 days. These mice, the total 21 mice, we euthanized them. We isolated their splenocytes, and we looked at the CD4 positive, CD44 high and 62 low, as well as the CD8 alpha positive, CD44 high and 62 low. The CD44 high, 62 low is suggestive of effector memory. And we saw a significant increase in the effector memory 65 days after they were re-inoculated compared to mice that otherwise have never seen the antigen. This was both on the CD8 alpha positive cells as well as the CD4 positive cells. So this to us was a bit encouraging, suggesting that there may be some sort of a memory effect to this particular antigen. So that was our preclinical work. And I just quickly give you a little bit of insight on the clinical work that we did using the Philips Sonaleaf HIFU system and comparing it with radiofrequency ablation. So we used the Sonaleaf system to thermally ablate certain tumors. As an example, we've treated desmoid tumors. We also were one of the first, along with our friends in Italy, across the world, to treat children with osteodosteoma, which is a benign bone tumor, which is otherwise, as seen here, which is otherwise often treated with RFA, as you can see, a RFA needle inserted, which is associated with quite a bit of morbidity, as well as these kids end up being hospitalized for at least a few days, require antibiotics and a lot of pain medication. Here, so here's an example of the osteodosteoma tumor showing neocortex with a nidus that tends to light up under contrast-enhanced MR. But after treating with MR HIFU, you do not see that enhancement anymore, suggesting that the nidus has been completely ablated using MR HIFU. So we place the focus and we heat it up beyond 65 degrees Celsius, confirming the loss of that nidus. The great part about osteodosteoma was if you treated the nidus, the tumor completely regresses, and the chances that it would never come back. But children with osteodosteoma, I mean, often take ibuprofen at least a couple of times a day, every single day, just to get on with their activities of daily living. So it was quite challenging and taxing to their body. So in short, the value proposition that MR HIFU and the thermal ablation brought to this was complete non-invasive, non-ionizing radiation approach, a day care procedure to treat osteodosteoma. And none of the kids had recurrence and pretty much went back in a couple of weeks without any more medication. So it's almost as if somebody were to see their skin or the region of treatment, nobody would think that there was any sort of procedure done. These are the different locations that we treated on the left side with MR HIFU. 3 of them in the femur, a few of them in the tibia, talus, calcaneus, and the phalanx. And with the RFA, we treated a couple in the upper arm, but pretty much most of them were in the femur and in the tibia. This was published in the Journal of Pediatrics a few years ago. So now I'm going to switch a little bit to imaging and give a flavor of what we're doing. So we also right now at IIT Hyderabad are running a couple of different clinical trials to quantify breast tumor heterogeneity in different tumor types using quantitative ultrasound imaging that includes shear wave elastography. So we, for example, use shear wave elastography. So the way it works is you create an ARF, or an acoustic radiation force. You focus your ultrasound beam, which then produces shear waves, which are transverse to the direction of your ultrasound beam axis. And then you calculate or you image the propagation of these shear waves. And based on the speed, you estimate your Young's modulus. So this is an example map of the Young's modulus of that particular tissue in that region. So what we set out to do is we collect volumetric images of the breast tissue. And then we said, we're going to have these images broken up as center of the tumor, to the left of the tumor and the right of the tumor, which are pretty much adjoining the center cross-section or the largest cross-section of the breast tumors. And we were very curious to see, apart from shear wave elastography, if there was anything else that could be picked up using ultrasound. And one of the things that we did was quantitative texture mapping of breast tumors. And GLCM dissimilarity is just one among a huge menu of these features. What you're looking at is invasive ductal carcinoma, DCIS, and invasive lobular carcinoma. Again, this was published last year. And I think, as per my understanding, the terminology has slightly evolved. We no longer classify them as IDC or ILC. They're all invasive breast cancer, I think. But please correct me if I'm wrong. So what we are showing here is the yellow or the orange region is the intratumoral region, and the blue is the peritumoral region that was captured about 2 millimeters away from the boundary of the tumor in the breast parenchyma. And what we're seeing is a significant difference in tissue texture between the tumor and the surrounding parenchyma. We also observe that in benign tumors, such as fibroadenomas. So here are 6 fibroadenoma patients, and we've looked at both the left, the center, and the right. They all tend to pick up certain differences, except this one patient. We right now have over 200 patients, of which about 100 of them are fibroadenomas, a lot of them already processed. And we have a better understanding why some don't pick up and some do. Hopefully will be online in the next few months. So then the next question simply was, is shear wave elastography sensitive? So we've been doing 3D shear wave elastography. And as part of that effort, the 3D shear wave elastography gives us the Young's modulus maps at different spatial locations in the breast tumor. So what we did was we picked up 2 regions of interest. We picked up inside the tumor. So the red circle and the yellow circle have some mean elasticity. The yellow corresponds to the blue line here, and the red corresponds to the red region of interest or the circle. So if you actually looked at 4 different patients, different regions of the tumor, elastography does not seem to think or suggest any change or difference in the Young's modulus or the stiffness of the tumor within the tumor boundaries. But our texture analysis for the same exact patients otherwise says there's something else going on here. So we think there's a significant difference in the texture of these breast tumors across multiple different parameters. Each row is a different parameter, and across these different frames, we are able to pick up a significant difference in the tissue texture. Now, what's already known is a lot of these tumors, breast tumors, are heterogeneous. We also understand shear wave elastography to a good extent to suggest why this may be happening. And we're hopeful in our upcoming paper that's something that we'll be able to explain at the best of our knowledge. So I'm switching gears very quickly. Very briefly give you a sense of sensors and tech in that space that we're developing and manufacturing at IIT Hyderabad in our lab at MURL. So we 3D print our own sensors, as you can see here. These are some scanning electron microscopes of the silver and the carbon nanoparticles that we use to pick up pressures and temperatures during ultrasound therapy. And what we've seen is our ability to map out change in the relative resistance because of how the sensors work to change in temperature. So this, in a sense, is a surrogate marker to a change in temperature. And that's essentially what we set out to characterize. And we were able to successfully do that. Each line is a single sensor, and we manufacture 23 of these to test them out. So again, very quickly, we also do a little bit of work on acoustic wave propagation. We create collagenous networks that are either randomly organized or have some sort of alignment. A good example is Achilles or patellar tendon collagenous networks are aligned. The fibers are aligned very well, almost parallel to each other. But in tumors, the collagenous network tends to be more randomly organized. Also, the fusion points or these red dots or the crosslinks is what we call them as cross-linking. The cross-linking can also affect how these waves propagate. And so depending on how we do our shear wave elastography and pick up the Young's modulus may suggest how the tumor's makeup is in terms of its collagenous. But the propagation of these shear waves are heavily reliant on these collagenous networks and their cross-linking density. And so our understanding of that cross-linking density and then subsequently the shear wave speeds will help us see if we can improve the 3D shear wave maps to further help our understanding in breast tumors. We also, sorry about that. We also did a little bit of work in understanding single nucleotide polymorphisms in breast tumors. We've created and come up with a framework that filters out different SNPs from breast cancer tumors. A trial that we've currently just completed where we've recruited about 25 patients who initially were treatment naive, diagnosed with triple negative breast cancer, and subsequently went through neoadjuvant chemotherapy, surgery, and radiation. We're currently preparing that report, and hopefully in a few months will also be online. But we found some very cool variants that were present in circulating blood that were otherwise not in the tissue, and vice versa. Also suggestive that certain things found in the blood may suggest a possibility of ovarian cancers in future. And so we're obviously not quite sure because those women still do not have any signs of that. But this is something that we are also reporting. In addition, we've also done some in silico work in trying to understand the effect of certain MEK inhibitors like binimetinib and against KRAS wild type or KRAS A146 type. And we've also seen what is their affinity and ligand efficiency. So just because it's an anti-KRAS, it does not necessarily mean it will do a great job. So that's something that we're trying to understand. And the reason we're trying to do all of this at a certain level is to see that when we plug in HIFU in the clinic, do we have a better understanding to what then HIFU does to this entire therapeutic regime. Thank you very much. I'm also thankful to everybody in my lab. These are some of our students. Some have graduated and left, and some are still around. I'm also thankful to All India Institute of Medical Sciences in Hyderabad, as well as Chennai Breast Center, with whom we're doing some of these breast cancer trials very closely for the last couple of years. We've collected over 300 patients worth of data, and our goal is to reach close to 1,000. And thank you again for your attention and time.
Thank you very much, Dr. Avinash. Excellent talk and very promising data. I'm looking at the chat box if someone has put a question. Otherwise, I can start with one question. One of the slides you showed was very heartwarming to see how this, in the pediatric population, how this HIFU is really making a difference. And especially with children, being off their their pain meds, and it's an outpatient procedure. So how, for these osteoidiomas, what's the regimen that you are doing? Is it a single day therapy, or is it over a course of several months or days?
This is pretty much for everybody that we treated in our pilot and pivotal trial. It was a single shot.
Oh, okay.
So they would come in the morning at around 6:30, go through regular check, get intubated, get treated. The treatment was anywhere between 2 to 2.5 hours. Extubate, pack you, go home.
Awesome. And do you give any pain meds? Because as you said, it causes some sterile inflammation afterwards.
Yeah, so all these kids get ibuprofen for the next week or 10 days. No opioids for any of these kids.
Dr. Josna Rao mentioned excellent talk. Is there any role in thyroid tumors?
So there's always a possibility with thyroid tumors, but the current standard of care for thyroid is, if you suspect that it's malignant, surgical resection is immediately done. Um, uh, we're trying to see if a non-invasive approach is possible. In terms of technical feasibility, it's absolutely possible.
Excellent. Any other questions from the audience?
I have a question, Saima.
Yes.
Hold on.
This is Dr. Milan.
Hi, Avinash. This is Milan.
Hi, Dr. Milith. Hi, Dr. Milith.
I'm at Memorial Sloan Kettering in New York City. Thank you for a very fascinating talk. And the question I have for you regarding your 3D shear wave elastography approach. You mentioned that approach has high sensitivity, but many of our approaches that are non-invasive tend to be lower on specificity. And so the question I had was what is the specificity with this approach, and whether there'd be concerns about overdiagnosis and overtreatment?
A great question. I think it's to put it bluntly, we still do not have the ROC curves for shear wave in varying breast tumors. I think people have reported changes in the shear wave maps. Where I stand right now, without any numbers that I'm able to show you, we are not quite confident that shear wave elastography, as it stands in 3D, is very specific in simply picking up a tumor type or saying a tumor is this type or this by Rad scale. There are issues in the way 3D shear wave is done using ultrasound, technically speaking. And we're trying to bridge that gap. So there's this company called Supersonic. They're originally based off in France. And so we just got into an agreement with them to get raw data access on the back end and trying to see if we can improve that. But yeah, I mean, as it stands, clinicians, at least in India, they don't do shear wave as a normal routine. It's mammogram followed by an ultrasound if needed. BMO.
Right. Okay. And then for your HIFU approach, what is the focal volume relative to the volume of the tumor that has to be treated? And then you may have said this and I may have missed it, but do you use a scanning approach or an approach to cover larger volumes?
Yeah, so I think you have two questions there. So I'm just going to sort of split that into two for my convenience. I'm sorry about that. But so what you see here is for the preclinical, I placed 3 foci. Each focus was about 7.5 to 8 millimeters along the beam axis, so along this, and it's about 2 millimeters across the beam axis. And I placed these foci adjacent to each other. Acoustically speaking, they cover maybe anywhere between 3 to 5% of the entire tumor volume, acoustically speaking. But then once you hit the tissue, you do tend to get a little more than where the acoustic pressures are, because acoustic pressures are not measured in tumor often. They're measured, in free field water, for example. So what is the right volume to be treated in patients? So there's a recent trial by Histosonics, which does cavitation histotrypsy, and they've done sparse therapy in liver tumors. And there's this whole thing about, is sparse therapy better than contiguous and so on. And some people say the Swiss cheese approach, where you have sparse therapy, seems to do much better. And they also seem to suggest that our trial at UMC Utrecht, we are doing a subtotal boiling histotrypsy with sparse locations. We don't know if that's good or bad. We don't know what volume to cover will produce the most favorable response. So that's something that we're still figuring out. I think.
Yeah, that's an interesting problem for optimizing our sampling.
Dr. Milind, there's 2 aspects to this. So technically speaking, can you reach every single location? So sometimes you can, sometimes you can't. And anatomically speaking, you may have locations that the tumor is sort of encompassing a major nerve or a vessel that you don't want to go too close to it. But can you still then produce a strong enough immune response that's potentially anti-tumoral?
Right.
So what is that? So you're absolutely right. I mean, it's interesting. We're trying to figure that out.
And then that brings up the other thought, which is, since you mentioned nerves, is whether you would use any contrast agents. Because right now, I think everything you're doing is label-free, right? As best I understand.
Everything is on the MR HIFU trial. We did pre and post therapy contrast just to make sure if the region that was not ablated was still alive or not. And so it's just confirmation of our region of ablation, basically to quantify non-perfused volume. But from a contrast standpoint, we actually just finished a trial of about 80 patients with breast cancer, looking at contrast dynamics and how they are related to tissue texture and stiffness and so on and so forth, and BIRADS and their outcomes in terms of therapy response. So that's something that we're actually writing up right now. We also have a label-free approach using basically a low-flow Doppler technique to map out intratumoral vasculature. A trial that we already started, recruited about 80 patients, and we're doing that volumetrically. So it's actually very exciting because we are now getting insights on how the the vessels inside these breast tumors are actually interconnected. It's actually really fascinating. Some of these patients have gotten neoadjuvant chemotherapy, 3 cycles, and came back, and we're seeing quite a bit of difference in the architectural change of these vessels. So again, very initial. I'm not quite sure where we're headed, but we're hopeful that that might help us. Again, label-free and contrast agent-free.
Yeah, great. Thank you. Good luck with all the work.
Thanks.
Excellent.
Hi, Dr. Eranki. I have a question.
Hi,
I'm Milan, senior scientist.
Okay. Hi.
Excellent talk, and so fascinating results. I was actually interested in understanding after the BH, did you see any stromal changes that would have driven the inflammation into the tumor?
The great point. We didn't stain for a stromal marker.
Okay, But would you
So,
expect or anticipate that would have you would have remodeled the collagen too in a way that would have helped the entry of immune cells?
From a acoustic standpoint, we know we disrupt anything that's in the path at the focus. So we know for a fact, even if there was stromal layer, that we would have disrupted it. But would that drive something into the tumor? I think that's a great question that we didn't look at. We didn't look at any sort of stromal markers. Yeah, I'm sorry, I don't really have the answer. And actually, that's very, very pertinent to maybe PDAC, pancreatic ductal adenocarcinomas, where they tend to have quite a bit of stroma. So then
Right.
we don't know that. So there's, I think, a couple of folks elsewhere in the world that we know are doing HIFU with pancreatic. There's one in Korea that's ongoing, but I'm not quite sure of what they're seeing in terms of how the stromal layer remodeling works.
Yeah, I mean, I was just curious to know because we, in Milan's lab, we are also looking at how stroma would affect ultimately what is infiltrating. So along those lines, I would have expected this to either bring about a remodeling of the fibroblasts that remain in areas that have not been ablated to bring about a change that would infiltrate. But this is something, in case you're interested to look at, maybe with staining. Maybe
Yeah,
this.
so what we
Right.
did, I think, was we looked at 11B positivity in a general region, not in the tumor, nothing specific. Again, it's a very, very unspecific or non-specific marker, right? In terms of, but yeah, we didn't do that. I think we definitely consider looking at that. And then are you also suggesting how this might also evolve from an epithelial to a mesenchymal or back or something maybe in the transition?
Not necessarily, but I would think that this would be critical to drive what you need, even for the immune checkpoint to work. You need the sentinels there, right? So this would be the one way they are driving. My related question was going to be my follow-up was going to be on the vasculature. You said that there was I don't know if you did any staining for the blood vessels too, because I would think then these are being modeled in the peritumor areas that remain, and then this is helping with the response.
Yeah, so on unrelated, when I say unrelated, is not in this study. On a different study, we actually treated mice at Utrecht along with a couple of other. It was a larger group. So what we found is for a certain set of parameters, vessels greater than 150 microns were still physically intact.
Okay.
Anything smaller than that were completely destroyed. So, but again, that's one set of parameters. So I picked a couple of parameters from that space when we started out. And we said, we do think that anything under 150, we're not quite sure if they were still structurally intact. But definitely ones that are 200 or 300 microns or bigger will stay intact.
I see. Okay. Yeah. I mean, this does help explain some.
Again, I mean, obviously would have been great if we had sectioned and looked at the vasculature. But yeah, we did.
Always in the future.
Always.
Yeah. Well, yeah, I'll follow your work, so I'll see what happens. Very
Hopefully.
interesting. Thank you so much.
Yeah, thanks a lot, Aditi.
Thanks, Aditi.
I'm Dr. Siddharth. I'm an endocrine surgeon from Apollo, Hyderabad. So excellent talk. I'm a clinician. So my interest is in ablating thyroid and breast tumors. So currently I'm doing microwave ablation for benign thyroids and even malignant thyroid tumors.
Okay.
Yeah, HIFU, I mean, I understand it's an expensive gadget to use in clinical practice. One. I would be interested to know what would be the lateral damage if you plan to ablate a particular zone, and what lateral extent, how many millimeters can we expect that it can get damaged?
I mean, it depends on, again, the parameters, but depending on how we tune it, if you want to just thermally ablate, and depending on how well perfused or non-perfused or poorly perfused the organ is, the peri-ablation zone can vary. So,
Okay.
so that's something that we can sort of discuss, and I can give you more specifics. But then, it depends on what's on the intervening tissue, how much acoustic pressures are deposited in the focus, and so on. To very quickly answer the expensive comment, you're absolutely right, especially if it's MR guided. So right now in my lab at IIT Hyderabad, we're the first in the country to develop a ultrasound guided HIFU system, which we're also sort of doing a very quick roadshow at AIG in Gachibowli this week, demoing it to them. So at some point, if you're interested to see, you're always welcome to IIT Hyderabad. We'll show you that. It's on a 6 degree of freedom robotic arm. And we've already have some pretty good data ex vivo as well as in tissue mimicking phantoms.
Great, thank you.
Thank you, Dr. Siddharth. I have a question from Dr. Sanjay Gambhir. Presently, what depth the HIFU probes are effective, that is for deep-seated tumors? Any data or immunostimulation in triple-negative breast cancer to make them sensitive to immune therapy?
Depending on the kind of transducer you use, you can go anywhere between a couple of centimeters all the way up to 13 to 15 centimeter depth. The clinical HIFU system that we used to treat osteoid had a focal length of about 13 centimeters. So shouldn't be an issue to reach liver. There was another set of researchers at UMC Utrecht who actually did treat the liver with multiple focal liver tumors. To the second question, in terms of the immune response in neuroblastoma, did I hear that right? I'm sorry.
Yeah, I
Dr.
think you.
Scott?
It was. I think I might to step down, so I'll take over.
Okay.
It was basically immunostimulation, to make them sensitive to immunotherapy. So basically, sensitize them to have a better effect with immunotherapy down the road.
Yeah, so that's basically what we show in this work. So we kind of show a little bit of what could have happened if we combine boiling histotripsy with an anti-CTLA4 and anti-PD-L1. So it does seem that boiling histotripsy is a strong adjuvant to immunotherapy. But again, we only understand so much. Right now, we're also doing clinical trials to see how that works in humans, because there's, again, a difference between how certain things work in murine models and how they work on humans.
Great. Another question from Dr. Gambhir is, presently to what depth the HIFU probes are effective, in reference to deep-seated tumors?
Yeah, so again, from a couple of centimeters all the way up to 15 centimeters is very easily possible and clinically possible today. Depending on the kind of transducer you use, the geometry, the frequencies, and so on and so forth. So it's very, very possible. The clinical HIFU system that we use to treat osteoid goes up to 13 centimeter depth.
That's pretty good. And basically, if we can do an anterior or a posterior approach, almost all portions of the body then would be accessible for the HIFU probes, correct?
Yeah, so the system was built to treat uterine fibroids. So I'm sure you can imagine that you have to go through the abdomen. Their patients oftentimes are prone, and they sort of lie down to treat the fibroids. So that's sort of originally how the system was built. So that's very easily possible. In fact, HIFU is in some sense. I'm just saying in some sense HIFU, but MR guided therapeutic ultrasound is FDA approved to treat essential tremor and Parkinson's today. So your ability to go through the skull non-invasively all the way into certain regions of the motor cortex.
And does it cause any damage to the tissue in between?
In between, you do not. So wherever the beam is not focusing, so in this region of the beam, you do not see any significant bioeffects. In fact, a lot of patients that we treated tens of patients in our previous clinical trial. None of them showed any changes to the skin or subcutaneous tissue or the muscle adjoining. None whatsoever. Completely intact. No effects even on the subsequent days. Only at the focus, because that's where.
What about bowel?
Bowel, again, it depends on how the beam is going through. So if you do have bowel gases, that may be a little bit of an issue. So we may have to
Okay.
come up with ways to fill that up with something else. So maybe saline or move them out of the way, if possible.
Awesome. I don't see any more questions in the chat. I had a couple of just philosophical or esoteric questions based on where the field is headed. It looks like this is being used mostly for tumors that are either localized and not easily amenable for surgical resection as of now. Because the standard of care for most early stage cancer is resection, unless there's a contraindication for surgery or the patient has comorbidities and other conditions that cannot undergo surgery and so on and so forth. So kind of the same rationale as RADONC was initially developed with SBRT and those kind of things. I think it's poised at that stage in the development to become a clinical modality that would be deployed in the clinic in the near future. Any thought process or any work that's going on to see if it can be applied to other stages of tumors?
So there's a couple of indications like prostate, liver, again, essential tremor, uterine fibroids, osteoid, so they're already FDA approved. Insurance now recognizes it. So at least in the US, there is reimbursement already for some of these indications to be treated with HIFU. So for such indications, I mean, you're looking at HIFU as an alternative to surgical resection or radiation. But then there are other indications that we're still exploring, metastatic cancers, where obviously the trial that we are running and others ran are on relapsed refractory tumors that are metastatic in nature, already metastasized. You've already exhausted your current standard of care options. Because that's obviously where you start. Right now, there's a huge number of indications that are both for the cancer and non-cancer as well. So essential tremor started obviously as a non-cancerous application. Parkinson's, looking at Alzheimer's, they're looking at mood disorders, ADHD, bipolar, and things of that sort. Now that's with the brain. They're also looking at opening the blood-brain barrier with ultrasound to improve the drug deposition in cases of gliomas and even glioblastomas. You also have applications in dermatology, cosmetology and dermatology applications. Again, non-melanomas and things of that sort. So I think in general, given the fact that it's non-ionizing and non-invasive, there's interest, but there's a lot of folks out there who are also putting in quite some effort to make sure some of these parameters are well understood before they can be deployed. And so that will obviously take its own natural time. So hopefully in some time, patients will have more options for treatment.
Yeah, one of the, I think it was Milind who mentioned about contrast. And I think you went towards MR contrast, but how about using ultrasound contrast, which can then act as a vehicle not only for localization but also for drug delivery and enhance the effect?
Yeah, so we right now, it's already been a year. We have a grant where we're trying to understand the effect of drug-loaded microswimmers. These are sort of active particles that can swim even against flow. So not like liposomes or nanoparticles that are loaded with DOCs, and the effect of HIFU to release them in certain regions of the tumor and not systemically. Our previous group has also done that. I was also part of a few experiments where we evaluated thermodocs and its release locally at the tumor location and not elsewhere. The lab that I worked with sort of predominantly did most of that work. So people have also looked at bubbles to initiate cavitation. So your thresholds to produce cavitation effects when you already have some bubbles in the region because you've injected some sort of a contrast agent reduces. So you don't need to use very high acoustic pressures to reach that cavitation threshold. So people have also demonstrated that possibility. I think where we stand right now is we want to see how it works without, get a feel for it before we combine. Again, on a completely different aspect, obviously that's not your question, but we're also looking at using SonoView. So for example, we're using SonoView to understand how these tumors are, but nothing really stops us per se in trying to see what happens if we then used HIFU as well with bubbles that were encapsulated with air or other gases.
Awesome. Yeah, I think that would be very exciting to if you have specific targeted dots or microbubbles, but that's for the future. I think one last question and then we'll wrap up. Where do you think AI fits into the? I mean, you alluded to texture analysis and a little bit of that, but in general, is there any work about utilizing tools that AI gave us for the space?
Yeah, so great question. In fact, with the data that we've been collecting, which is really cool, so we have patient-matched 3D ultrasound with 3D mammo, with immunohistochemistry and other patient information. We're now getting fairly close, and we're also doing contrast and vasculature mapping in 3D. We're now getting a little closer to sort of gently suggesting the BI-RADS score to the physician or radiologist or the sonographer upfront. The hope is that we'll also be able to pick up lesions that are benign or malignant even before the biopsy. Again, these are all suggestive. We're not trying to say we're replacing biopsies, but certain information, especially in BI-RADS 4A, 4B versus a 3, can sometimes be questionable. They could be downgraded or upgraded sometimes. And so ability to sort of use these tools in a slightly lower resource setting can potentially be helpful. And that's something that we're kind of working on already.
Awesome. Looking forward to some great work from your lab here in the US as well as in IIT Hyderabad. So thanks for a great talk. I think we'll close the session. The excellent discussion down the road. So thank you, Avinash, for the great talk. And we will have our next session next month, same time, same platform. See you then. Thank you, guys. Enjoy the rest of your weekend.
Thank you very much.
Thanks. Bye-bye.